Hi, thank you so much for watching this video today. My name is Sarah Sagui, and this is Lydia Roos.
Hi.
We are Health Psych PhD students here at UNC Charlotte, and today we're holding another virtual review session where we're going to cover the material for Exam 2 in the Psych 2113 Introduction to Brain and Behavior course. This video is going to be about 45 minutes to an hour long, and we hope to cover some of the more important and difficult aspects of the exam. And before we get started on the material, we wanted to review the format of the exam again. We actually discussed this in our previous virtual review session for Exam 1, but we wanted to reiterate it again for those students who are just starting to watch these videos now. So the exam is approximately 60 questions long, and it's going to consist of true, false, multiple choice, and matching questions. So keep in mind that some of these matching questions in particular require multiple answers. So within one question, you may need to match 5 or 6 concepts. The exam is going to be posted online for 72 hours total. And the start of that window is listed on your syllabus, and Dr. Levins will also have made that clear in her lectures. So it's open for a 72-hour window, but once you begin the exam, you only have 1 hour to complete it. So keeping in mind this approximately 60 question length, that gives you around 1 minute per question. So most students finish on time with no problem, answering all the questions and doing well. However, some of the students have said that it's kind of difficult to complete this online exam unless you're really well prepared for it. So we just wanted to take this opportunity to remind you that you're not going to have time to look through your book or look through your lecture notes or other online materials while you're taking the exam. You really need to be well prepared and ready to answer each question from memory when the exam's open because you only have an hour. So just a reminder that an online exam doesn't mean that it's an easier exam. In some ways, it can actually be harder. And these exams in particular are very rigorous and test your knowledge of the material. So it's important that you take the exam at a time when you feel really awake and alert and possibly in a work type environment. So maybe sitting at a desk with your computer in the morning when you're feeling awake and alert.
This exam is going to cover Chapter 6, which covers the hormones, as well as the posted collat chapters on hormones. So that's the 2 chapters called Chapter 11 and 12, and they're uploaded as PDFs on Moodle. And then there's also going to be Chapter 7, which covers the way that we study the brain's structures and functions, as well as Chapter 9, which covers how we sense, perceive, and see the world. And then Chapter 10, which reviews how we hear, speak, and how we process language and music. And then Chapter 11, which reviews how the nervous system responds to stimulation and produces movement. So again, we're not going to go over all of the information in great detail. You do have the detailed study guide, so we're just going to go over all of the more difficult or more important aspects of the exam and what you need to know.
Okay, so we're going to start with Chapter 7, which talks about the different ways that we study the brain's structure and functions. So we've already discussed many aspects of our brain and our behavior and how those things interact. But it's also important to know the main techniques used to study and measure how the brain works and how it influences behavior. So for the exam, it's important for you to understand the overarching ways that we study the brain and the specific mechanisms we use to accomplish this. So for example, in the field of behavioral neuroscience, which studies the biological basis of behavior, they use techniques like neuropsychological testing, behavioral analysis, brain lesions, and even brain stimulation. And we can also study the brain and behavior using EEG recordings and MEG, and that's to study the electrical activity of the brain. We can also do static imaging, which are things like CT scans and MRIs. And then we can do dynamic imaging of the brain, like fMRIs and PET scans. And then finally, we can look at the brain chemistry and genes and gene expression using various techniques. So it'll just be important for you to know these general techniques and the types of information that each of them provides. This is information from your textbook that's in the beginning of Chapter 7. Okay, so that's broad information. Now we're going to briefly review sensation and perception because it's going to help us organize the rest of the material about vision and auditory processing and about movement and somatosensory processes. And this will be especially helpful because there's an overall pattern for each sensory system that we're going to return to as we're explaining those. So there are specific receptors for each sense and a primary cortex area where that information is processed. So really quick, looking down at this figure right here, there will be different receptors for each system, and then they are translated into neural messages and sent to a specific cortex that relates to that sensory system. So sensation refers to the registration of physical stimuli from the environment by the sensory organs. So this diagram is labeled generically here, but it can be used for each of the systems we're going to discuss. So essentially, we move through these processes, and sensation occurs when external information from the outside world enters the specific receptors. And so here we can think of this entire process as sensation happening. So when we encounter this sensory information, for example, maybe we're driving at night and we see a bright light, this is going to enter specific receptors in our visual processing system. Then these receptors convert that environmental energy or that light into neural activity, which is a message for the brain. This information is going to travel through some sort of thalamic relay nuclei and onto the primary and secondary sensory cortices, and then finally the association cortex. And as we can see here, when that information travels to these cortices, this is considered perception. So because our sensations are affected by context, including our emotional states and our past experiences, we refer to perception as our subjective interpretations of these sensations by the brain. So make sure that you know this generic pathway and the difference between sensation and perception and how those interact. So using this information, let's begin with our first sensory system, which is visual processing. So as an overview, visual processing occurs when light energy from the outside world enters the pupil, which goes into the eye and hits the light-sensitive surface in the back of the eye called the retina. This is going to stimulate photoreceptor cells on the retina, which begins to create a visual world for us. Regarding the visual pathways into the brain, remember the visual pathway is the same as the diagram we just went over, but it's specific to vision. So first, photoreceptors detect light, and we're going to go into more detail about photoreceptors in a moment. And then the retinal ganglion cells form the optic nerve, which serves as the pathway to the brain. The junction of the optic nerves for each eye is actually called the optic chiasm. And here, axons from the nasal halves of each retina cross over to the opposite side of the brain, but the axons from the temporal halves remain on the same side. So this means that information gets crossed over, and that visual information from the left field goes to the right side of the brain, and vice versa. So from the right visual field goes to the left side of the brain. And then after these messages reach the brain, the visual information goes through the thalamic relay nuclei, like the diagram we looked at above, and it specifically goes through the lateral geniculate nucleus, or the LGN. After that, messages are sent on to the occipital cortex, which consists of the primary visual cortex and the secondary visual cortex. So these are both important to know. The primary visual cortex is labeled V1 and is also called the striate cortex. And this receives the input from the LGN, which is in the thalamus. And from here, two different pathways emerge to the parietal and the temporal lobes. So what we call the dorsal visual stream is the pathway from the occipital cortex to the parietal cortex. And this represents the how pathway, or how we're supposed to basically act toward different objects. The ventral visual stream is the pathway that goes from the occipital cortex to the temporal cortex, and this is known as the what pathway, which identifies what an object is that we are looking at. In the secondary visual cortex, which is labeled V2 through V5 and also called the extrastriate cortex, this is an area that represents the visual cortical areas outside of the striate cortex. So that was the visual pathway to the brain, and it is important for you to understand how we see color, which is sort of this next section here, but I won't review that. Instead, I'm going to talk about the structure of the eye because this can be confusing to students sometimes. But make sure you know about color blindness and these two different theories about how we see color. Okay, so moving towards the structure of the eye, starting on the outside of the eye, we first have the cornea, which is a clear outer covering of the eye. Then we have the iris, which opens and closes to allow more light in, or less, depending on the circumstances. And the hole within the iris is actually your pupil. Then we have your lens, which focuses light, and it can bend to accommodate near or far objects. So when you're thinking about nearsightedness or farsightedness, you're thinking of your lens, which can bend accordingly. And then it's not until the retina where we actually see the light energy initiating neural activity. So this is an area at the back of the eye that is very light sensitive. And it's because there are neurons and photoreceptors here that translate light into action potentials. So in the retina, we have a couple of different things to consider. We have the fovea, which is the center of the retina for high acuity, meaning that our vision is better in the center of our visual field than at the margins or periphery. And again, this makes sense because the point that you're staring right at is going to be the most accurate or the most highly acute to see, whereas things in our periphery are more difficult or can be a little bit blurry sometimes. And then we also have a blind spot. So this is an area that has no photoreceptors because it's the optic disc in the retina where the axons actually leave the eye and the blood vessels enter and leave the eye. So light comes in and it hits the retina, which initiates neural activity by stimulating the photoreceptors. So there are two main types of photoreceptors, rods and cones. Starting with rods, there are actually much more rods than there are cones in the eye. And these photoreceptors are sensitive to low levels of light, and they come into play when we need night vision or motion detection. So, for example, when you're camping at night and you're walking around in the dark, your rods are active because you need to be able to see in the dark at low levels of light. And that's why you can still see at night, but everything is sort of one color because there's only one type of pigment for the rod photoreceptors. Cones, on the other hand, are actually much more responsive to bright light and they process color for us. So one thing that helps me remember the difference between rods and cones is that cones starts with C and so does color. So cones see color. And cones are mostly located in the fovea and they help mediate color vision and our ability to see fine detail. And unlike rods, cones actually have 3 different pigments and any given cone has one of these 3. So they absorb light over a range of frequencies that we can see here for blue, green, and red. And here are some diagrams taken from your textbook that describe in a little more detail or visually what the cones and the rods look like and how light enters the eye and is translated into a neural message. Okay, so moving on to our second sensory system, which is auditory processing. I'd like to go over this section in a similar format as the last section. So first I'll talk about the auditory pathways to the brain, and then I'll go into some detail about the structure of the ear. So it's actually organized a little bit differently on this study guide. So we're going to scroll down. And we'll come back to this in a moment, but we'll start here. So on a basic level, the auditory system converts physical properties of sound wave energy into electrochemical neural activity that then travels to the brain. So recall that all of the sensory systems follow that same pattern, where the receptors take in information from the outside world and then turn that into a neural message, which goes through the thalamic relay nuclei and then on to the primary and secondary sensory cortices and onto the association cortex. So in the ear, the auditory receptors are in the inner ear, and we'll talk more about where that is and the structure of the ear in a moment. So it's actually hair cells within the inner ear that transform sound waves into neural activity. So there are about 3,500 inner ear hair cells, which are the auditory receptors, and there are about 12,000 outer hair cells, which alter the stiffness of the tectorial membrane, which is the membrane that stimulates hair cells. So when the inner hair cells are stimulated by waves in the cochlear fluid, the outer hair cells generate graded potentials in the inner hair cells, which stimulate that neural message. And then the neural messages for sound travel from the ear to the thalamus. The axons of each auditory nerve synapse in the cochlear nuclei project to the superior olives on both sides of the brainstem. And then these axons project to the superior colliculi, followed by the inferior colliculi. And then finally, axons project from the inferior colliculi to the medial geniculate nuclei of the thalamus, which is our thalamic relay nuclei for the auditory processing system. The lateral and medial superior olives do react to differences in what is heard by the two ears, so it's important to know that the medial reacts to arrival time differences and the lateral reacts to amplitude differences. Then from the thalamus, the neural messages go to the primary auditory cortex, and these auditory signals are conducted in two areas of the association cortex. So these are important to know. The anterior auditory cortex identifies what a sound is, while the posterior auditory cortex locates the sound or where that sound is coming from. So that was sort of an overview of how sound travels from the outside world into our perception. Make sure you also study the other aspects of this chapter, like the properties of language and music as sounds. But for the remainder of this section, I want to talk about the structure of the ear. So we're going to scroll back up here. Okay, so we tried to make this pretty detailed and in-depth for you by putting some of the key figures from the book and from your lectures in with the study guide. So essentially, like it says here, the ear collects sound waves from the surrounding air and converts that, what we would call mechanical energy, into electrochemical neural energy, which is those neural messages that are being sent to the brain. These messages are routed through the brainstem and to the auditory cortex. And so talking about the structure, your book says that the ear is a biological masterpiece in 3 acts, consisting of the outer ear, the middle ear, and the inner ear. And so we're going to walk through those 3 sections of the ear right now. So the outer ear structure includes 2 main features, the pinna and the external ear canal. So where we can see right here, the pinna is this funnel-like external structure, and it basically catches sound waves in the outside environment and deflects them or channels them into the ear canal. And then this external ear canal right here amplifies those sound waves and sends them into the eardrum, which vibrates depending on the frequency of the sound wave. And I just want to make a note here that make sure you know information about sound waves, including the physical properties of them and things like frequency, amplitude, and complexity of sound waves. Okay, so for the middle of the ear, this is an air-filled chamber that holds the ossicles in it. And the ossicles are bones in the middle ear, in this air-filled chamber. And there's three of them. They're called the hammer, the anvil, and the stirrup. And they are actually named this way because of their distinct shape. They actually look like the objects that they were named after. And these connect the eardrum to the oval window of the cochlea, which is located in the inner ear. So now we're starting to see how the structures of the ear actually fit together. And so each of these has a unique purpose. So make sure you know in some detail the purpose of each of these smaller structures. And then finally getting into the inner ear, which we sort of touched on already, talking about our hair cells, the receptors for the auditory system. In the inner ear, we have the cochlea, which is a fluid-filled structure that holds those auditory receptors. And we call this the organ of Corti, which has both the cochlea receptor cells and those other cells that support them. Also in the inner ear, we have the basilar membrane, which is this receptor surface on the cochlea that turns sound waves into that neural activity that is then processed within the auditory cortex.
Okay, so now we're going to move down to movement. I know that this isn't quite in order, but we're just going to start here because it'll make the rest of it make a little bit more sense, I think. So first of all, just make sure that you're comfortable with sensory and motor patterns. You can think of it as either top-down or bottom-up processing, with sensory starting from the bottom with muscles. And so it's going through the brainstem motor nuclei to the primary cortex and the secondary motor cortex, and then finally to the association cortex. But the motor pattern, which is opposite from sensory patterns, start from the top with the association cortex and move all the way down to the muscles to perform movement. So remember that sensory pathways are afferent, while motor pathways are efferent. And also just make sure to remember that extensor muscles extend the limbs, while flexor muscles bring the limbs towards you. So you will need to know that the basal ganglia and the cerebellum interact with different levels of the sensory motor hierarchy to coordinate responses, and also try to be familiar with the types of motor symptoms, so that's hyperkinetic and hypokinetic, and the kinds of effects that they produce. Do make sure that you know the major components of the motor system, as well as how the forebrain helps to initiate movement, including knowing the roles for the different frontal lobe regions. So moving down to corticospinal tracts, these are the main efferent pathways from the motor cortex to the brainstem and then to the spinal cord. They include the lateral and ventral corticospinal tracts, which operate the limbs and digits and trunk muscles, respectively. So the corticospinal tracts stop in the spinal cord, where cortispinal fibers make connections with interneurons and motor neurons. The interneurons project onto motor neurons, and the motor neurons send information through the axons to the muscles to produce movement. And as I mentioned earlier, the sensory pathways are afferent by taking inward through the somatic nervous system, while motor pathways are efferent and send information out through the central nervous system. And also make sure that you understand the homunculus and the topographic organization, which are the representation and organization of the body in the sensory or motor cortex. So in this, the body parts that require more attention for intricate motor control, like the facial muscles or fingers, or things that are more sensitive to touch, like lips or hands, they have larger representation in the cortex. So now we're going to go back up in the study guide to somatosensory pain and vestibular system. All right. So the somatosensory pain and vestibular system. So the somatosensory system tells us what's going on with our bodies and in the environment through perceiving sensations like touch, temperature, pain. So the different kinds of somatosensory perceptions include nociception, hapsis, and proprioception. So nociception, which is how we experience pain, temperature, and itch, is perceived by free nerve endings activated by chemicals. So you can remember this because nociception comes from the same Latin root as noxious, which means harmful or damaging. And pain and temperature and itch can all be damaging, or at the very least, they can be obnoxious. So nociceptive information is carried through the ventral spinothalamic tract. Hapsis is how we perceive fine touch, pressure, and vibration, and is activated by stimulation of things like hair and tissue. And proprioception is how you perceive the location and the movement of your own body. So you can remember this because of the Latin root of proprioception is proprius, which means one's own. So it makes sense that proprioception is about perceiving your own body. So both haptic and proprioception information is carried through the dorsal spinothalamic tract. And I also recommend going over the ways that unilateral spinal cord damage can affect hapsis, proprioception, and nociception in the body. So I'm going to skip over the details for the primary and secondary somatosensory cortexes right now because Sarah's already covered that a little bit. But just remember that the receptors receive the sensory information, which then travels to the thalamus and then to the somatosensory cortex. So going down to pain, pain is definitely necessary in life, and someone who can't experience pain would not have much to make them avoid harmful situations or injuries. So the gate theory of pain is based on the theory that pain signals enter neurological gates at the spinal cord, and whether those gates are open or closed determines whether the pain signal actually reaches the brain or not. So these gates can be closed if you're concentrating on another activity. So that's like when your doctor asks you about your schoolwork while you're getting a shot to distract you, and it makes the shot seem like it hurts less. So it's also influenced by other sensory information. So if you're experiencing pain through nociceptive stimulation, but then you're also experiencing haptic proprioceptive stimulation, the haptic proprioceptive stimulation can actually close the gate that the nociceptive information would have normally traveled through. So this is why it makes it, feels better when you rub or press on a body part that you just hurt, because it's blocking the other pain information from traveling. So the last thing within the movement chapter that you'll need to know is about the vestibular system, which is a somatosensory system that allows you to be able to sense the position of the body in relation to gravity and the movement of the head by sensing the direction and the speed of head movements. Okay, so now we're going to go back down. So now we're going to go into the hormones. It covers pages 202 to 210 of Chapter 6 and the GLAT chapters that were posted on Moodle. So hormones are different than neurotransmitters, which are also called neuropeptides. While neuropeptides can travel through the nervous system, hormones travel through the circulatory system, also known as the bloodstream. So this means that even though hormones and neurotransmitters can be started at the same time, the effects for hormones don't always occur right away. So they can also last longer in our bloodstream. The endocrine glands are listed on the study guide, so I'm not going to go over each of them in detail, but so you have that information as well. But I do want to go in a little bit more detail about the pituitary gland. So remember that there is both an anterior and posterior portion of the pituitary gland. The anterior portion is truly a gland, and it produces hormones as well as stores them and releases them. This portion is responsible for adrenocorticotropin hormone, or ACTH, the growth hormone, thyroid stimulating hormone, luteinizing hormone, and prolactin. The posterior portion of the pituitary gland, on the other hand, does not actually produce hormones. It just stores and releases oxytocin and vasopressin that are produced by the hypothalamus. So the way that hormones are transported through the body is through the hypothalamus first, at least for those hormones that are created there instead of the pituitary gland. The neurohormones are produced and then they stimulate the pituitary gland. The pituitary gland then releases the hormones to influence targeted endocrine glands. The endocrine glands release the appropriate hormones, which then travel through the circulatory system until they find their target organs and tissues to either stimulate or inhibit their function. That gives you a little bit of a better detailed picture of how all of that works. There are two classes of hormones. So that's peptides and steroids. Peptides are chemical messengers that are synthesized by cellular DNA. And they act to affect cell physiology, but they require a second messenger to do this. And peptides are things like insulin and the growth hormone, while steroid hormones are fat soluble and are synthesized from cholesterol, and they interact directly with DNA. These include gonadal and thyroid hormones. So there are these two classes, but then there are also three main functional groups of hormones. So I'll talk about those a little bit. So one group is the homeostatic hormones, which maintains metabolic balance and allows our bodies to self-regulate to remain homeostatic. So for example, insulin regulates the amount of glucose in your system to maintain homeostasis in most individuals. This can be dysregulated when cells become resistant to insulin and the body can't make enough to keep up. So another example is melatonin, which operates on a circadian rhythm, which means it differs throughout the day. So at night, it rises and induces sleepiness, and it's homeostatic in that it makes sure that we get the rest that we need every night to maintain balance in our systems. There's also gonadal or sex hormones and glucocorticoids, which I'll go over both in more detail. So going down a little bit. So gonadal hormones control reproductive functioning as well as sexual development and behavior. They affect us before birth and continue throughout life. According to the organizational hypothesis, the actions of these hormones, so the timing and the amounts during sensitive developmental times, can affect personality and sexual orientation. And the levels of hormones in utero can be influenced by things like genes, stress, and the mother's immune system. So within gonadal reproductive hormones, there are three main types. So that's androgens, which include testosterone, estrogens, which include estradiol and progesterone, and pituitary hormones, a few of which we talked about earlier. So keep in mind that these hormones are sexually dimorphic, which means that both males and females produce all of these hormones, but one sex might produce a great deal more than the other sex. So now we're going to move down to stress hormones. So remember that a stressor is something that induces a stress response. Physiologically, we respond to stress with two systems that start at the same time. So that's the autonomic nervous system and the hypothalamic pituitary adrenal axis, more commonly known as the ANS and the HPA axis. So since the ANS utilizes the nervous system, effects are almost immediate, while the HPA axis takes longer before you see these effects, and that's because it's controlled by the hormones, so they travel throughout the bloodstream, the circulatory system, so they're not immediate. They might start at the same time, but you don't usually see the effects until a little bit later. So it's not quite as immediate as the ANS. So the HPA axis is what produces cortisol, which is one of the most well-known and well-researched stress hormones. It's released by the adrenal glands in response to a perceived stressor, and it's released so that we can break down proteins and to increase the amount of glucose in the blood to get us kind of ready for action. So this is where Scroll down a little bit. This is where allostasis and allostatic load come in. So in response to stress, our body works to regain balance and homeostasis through physiological and behavioral changes. So the short-term adaptation is called allostasis, and it is adaptive. However, if someone continually goes through chronic stress or a lot of repeated stressors, the body's attempt to adapt over and over again when recovery either might not occur, or if it only occurs for a short period of time and it doesn't really allow the body to kind of rest and recuperate, this can lead to long-term cumulative effects. So this is what's called allostatic load, and that can be damaging to physiological symptoms or systems. Excuse me. There are some gender differences that could influence stress responses. The hypothalamus is larger in men, which might be a reason for their greater cortisol response, along with their greater amounts of testosterone, vasopressin, and estradiol. And then the tendon friend theory was created to explain some of the differences in stress response behaviors among males and females. The theory states that since females have in the past been very maternally invested in their offspring, it doesn't really do much good for reproduction if the next generation, or for the next generation, if everyone just runs or fights at the first sign of stress. So in females, it's hypothesized that in order to better protect their young and themselves in times of conflict, they actually produce more oxytocin to enhance their abilities to tend to others and to maintain their social networks, and reduce their HPA axis response to stress to hopefully help along themselves as well as the next generation.
Our virtual review session for exam 2. We just wanted to reiterate that the exam will be open for a 72-hour window, and once you start the exam, you have 1 hour to complete it. So make sure that you're really well prepared at that time that you sit down to take it. Make sure you're in a good study environment, and again, you won't really have time to flip through the book or your notes while you're taking it. So just ensure that you've used all of the materials provided to you. You have access to all of the lectures online. You have the detailed study guides, the book, and your own notes, as well as this review video. So please utilize everything and use it to your advantage to do well on this exam. So